T cells are an important pillar of the human immune system. They can mount a strong response against viral infections, and they can recognize and eliminate cancer cells. However, often cancer cells have acquired mechanisms to escape the immune defense. This is a huge problem. To solve this issue, one needs to find out which specific antigens are expressed only on the cell surface of cancer cells. CAR T cells are modified to detect these antigens. Like regular T cells, they possess normal T cell receptors. Additionally, specific chimeric antigen receptors are expressed artificially. Similar to the key lock principle, CAR T cells perfectly match the tumor antigen, and the T cell's cytotoxic mechanisms take care to eliminate these cancer cells. The only characteristic that makes CAR T cells different from natural T cells is the chimeric antigen receptor. But how are CAR T cells made? In step number 1, blood is removed from the patient and the T cells are isolated and cultured. Step 2 involves the genetic modification. For that, one can use a viral vector such as the lentivirus. This virus carries the genetic information that encodes for the chimeric antigen receptor. In a process known as viral transduction, the T cells are infected with the lentivirus, which inserts the CAR gene. Following transcription and translation, the protein receptor is produced and the patient's T cells are now equipped with tumor-specific chimeric antigen receptors. In the next step, the cultured CAR T cell population is expanded in the lab and then finally infused back into the patient. Inside the patient, the CAR T cells patrol the whole body. Once they encounter tumor cells with the distinct antigen, such as CD19, they recognize the cancer cell, and the altered conformation of the receptor activates downstream signaling. There are many different ways how CAR T cells can eliminate the cancer cell. A common mode of action is the release of cytokines, which kills the cancer